The overall goal of the following experiment is to analyze the extent of cell-to-cell movement of fluorescently tagged protein in plant epidermis. This is achieved by obtaining DNA-coated gold particles to express the fusion protein of interest transiently in plant epidermis. As a second step, the DNA is delivered into the plant epidermis, which allows the expression and movement of the tested fusion protein. Next, the expressed fusion protein is observed under a confocal microscope in order to monitor the expression and localization of fluorescently tagged tested proteins. Results are obtained that show the extent of cell-to-cell transport of the fluorescently tagged protein based on analysis of the distribution of the fluorescent signal by confocal microscopy.
Hi, I'm Ben Meyers from the laboratory of Citovsky in the Department of Biochemistry and Cell Biology at the State University of New York at Stony Brook. Today we will show you a procedure for analyzing extent of cell-to-cell movement of fluorescently tagged protein in plant epidermis. We use this procedure in our laboratory to study cell-to-cell transport of plant viral movement proteins in various plant backgrounds and to compare the cell-to-cell mobility of different types of proteins. So let's get started.
To begin this protocol, grow one Arabidopsis plant on ProMix BX in an appropriately sized pot. Use an environmentally controlled chamber with a short photoperiod and 40 to 65% relative humidity. For Nicotiana benthamiana and Nicotiana tabacum plants, grow each plant on ProMix BX in a larger pot. Once again, provide an environment-controlled chamber with a long photoperiod and 40 to 65% relative humidity. Fertilize the plant occasionally with commercially available products as previously described.